One treatment using Exon technology is the Dunckley experiment. To begin their research, they needed to address how they were going to skip over that exon 23 that had the stop codon here in the left diagram. The answer was an antisense oligonucleotide being placed prior to the stop codon that also needed to be binded to the mRNA. Figure 1 shows where this needs to occur. The next issue was how the antisense oligonucleotide was going to get into the cell. Figure 2 to the right here displays the execution of how they're going to package the antisense oligonucleotide. They package them through PEI. This was shown effective through immunostaining here. Here's 24 hours, and here's after 48 hours. The overall question is now, how can translation be restored in the dystrophin protein as a possible treatment for patients with Duchenne muscular dystrophy? The researchers' hypothesis was that the dystrophin protein can be restored using a complementary oligonucleotide sequence of a mutated dystrophin gene. The experiment begins by using the ideas answered earlier in figures 1 and 2. The left diagram is the PEI that is packaged with the antisense oligonucleotides. And these are now being placed onto specific cells. These cells are from the Bullfield 1984 MDX mouse. Figure 3, this image right here, explains how the process of immunostaining worked. For here, part A, the green part is how the antibody, and how this worked is here's the primary antibody that the goal is to have that bind to the possible dystrophin protein if there was anything, and everything was washed off, and if anything that didn't stick, it wouldn't be there, the presence wouldn't be there. And then on the second image here, there is the primary antibody that the goal is to have that stick to the dystrophin protein, and then the second antibody is now added to attach to any possible first antibodies. And immunostaining is used on the second antibody to see the glowing cell, to see if it was attached at all. Figure 3A on the left displays the results of cells where the first antibody was added, but it didn't bind anything since there was no dystrophin to bind to. In figure 3B, the results were treated glowing cells where the first antibody was attached to the dystrophin and the second body was able to attach. So you'll see the presence of glowing cells on the dystrophin protein. To conclude, the experiment supported the hypothesis. The results from 3B show that there were an exon skipping treatment through the displayed production of dystrophin. However, a clear caveat is that only 1% of the MDX mouses treated displayed these results. A majority showed the results in A. Only 1% showed the glowing cells on the dystrophin protein. Another caveat would be, due to such little effectiveness of this treatment, only 1% showing to be effective, it would be very costly to have this treatment be effective on real patients with Duchenne muscular dystrophy. However, overall, the experiment appeared to show that exon skipping technology can be effective. Figure 3A figure 3B, I mean, shows that the results of the first antibody was able to attach to the dystrophin. There are glowing cells.